So this lecture is about vaccines, antivirals, and immunotherapy. And we went through a bunch of this, or we started to go through this in class, so I'm going to quickly go through the first few slides. Remember that the point of a vaccine is to produce long-term memory of the immune system, so that when a person is challenged with the actual, in this case, virus, their immune system is primed and can attack or knock down the viral infection faster to prevent illness. We talked about the vaccine schedules. This is for kids 0 to 18 years. This is for people over 19. And we compared what is recommended now versus what was recommended a little over 30 years ago. So quite a difference. We've talked a little bit about the positives and negatives of vaccines. Unfortunately, like I said, we just don't have time to get into that here. We talked about the history of vaccines. So this was to prevent smallpox. And a similar virus called cowpox was actually used to inoculate a little 8-year-old boy who was then later challenged with smallpox and shown to not get the disease. So hence our first actual vaccine trial. And again, this is showing the goal of vaccines. And right here we're looking at antibody levels. So remember, antibodies are specific to what we call an antigen. And in this case, it would be the outside of the virus. And so the idea is that you have a low level of antibodies circulating, kind of keeping an eye out for another virus, and once that virus once you get an actual infection, the antibody level can go way up and help knock down this virus infection. Antibodies are made by B cells, and what we're talking about right here is memory. So the goal of vaccines is memory, immune system memory, and a lot of that is due to these little guys called antibodies. We can also get memory with T cells. And it takes, this is part of what we call our adaptive immune response, which is a specific response. So there's an innate immune response, which are things that are non-specific. So anytime something foreign comes into the body, the innate immune response kicks in first, and then it takes about a week to get the adaptive immune response going. So you can imagine why, if we can have a vaccine that stimulates the week waiting period for that adaptive immune response, and then it's hanging out with memory, so that the second exposure you have a much greater, much faster immune response. An important concept with vaccines is the idea of herd immunity. Herd immunity means we know that not 100% of people are going to be immunized. Okay. But the more we can get immunized, which is all the little yellow people, the better the chance we have to break the chain of transmission and prevent people from getting sick, even those that are not immunized. So up here it's showing no one's immunized. We've got a couple sick people, and the disease just spreads through the population. Here we have a low level of immunization. Those people are going to be protected, but the rest of the non-immunized people will not. And in this is where herd immunity comes in, is that most of the people are immunized. You get a few sick people, but if you can see here, only one additional person got sick because we're protecting these other people by this herd, so the virus in this case can't spread to this little blue guy because everybody else is vaccinated. So that's the idea of herd immunity. It's getting more important here in this country as more people are opting out of vaccinations. Really important in developing countries where sometimes it's hard to reach every person to get them immunized. So let's talk about some types of vaccines. In this slide, I'm just going to give you a quick overview, and then on the next slides, we'll talk about the advantages, disadvantages. So an attenuated live virus, so we're talking about viral vaccines, is basically you take a virulent strain or a virus that causes disease and you make it avirulent. That means it cannot cause disease. And this is caused by mutation. And so what happens is you grow the virus in less than ideal conditions. Ideal conditions. And the virus will, So you grow the virus in less than ideal conditions, and the virus will start accumulating mutations to adapt to that new host cell. Okay. So then they're going to accumulate mutations that make them not cause disease in the human host. One interesting note about the influenza vaccine is that it's normally grown in chicken eggs, and the virus mutates in the chicken eggs because it's trying to adapt to growing in this non-natural host. And so there's more mutations, and what they're now discovering is that this may not be the ideal way to grow influenza vaccines because those mutations are actually making the virus vaccine, I should say, less effective for humans. So they're actually now looking at switching growing influenza from chicken eggs into mammalian tissue culture. So like in a petri dish, and they're finding that the vaccine virus is much more stable, less mutations, which they believe will make a better vaccine. All right, so the other type of using a whole virus is an inactivated or killed. So you chemically or use heat basically cross-link, I should write that word out, proteins, a lot of times the capsid, and or genome, so nucleotides. And you cross-link these chemically or with heat so that they cannot perform their function. Okay, so if you cross-link the capsid, maybe it can't open and release the genome and start the replication cycle. Or you do something to the genome so that it cannot cause disease. So these do not cause disease. Okay. But they look like the outside of the virus, so they're good at stimulating an immune response. Subunit means you just use part of the virus to stimulate immunity. And you're using part of the outside, you're using sometimes just protein pieces, sometimes a virus-like particle, we'll talk about in a minute. The advantage is there's absolutely no possibility of disease because there's not a whole virus being used for the vaccination. So, whole virus, attenuated, live. Let's talk about the positives and the negatives. So the advantages, disadvantages of this type of vaccine. So the biggest advantage is that this virus does replicate, and that makes it best for stimulating a strong, long-lasting immune response. Strong, long-lasting immune response. So it is a weak virus, and so your body is reacting to it just like it would a regular virus. Many times you can decrease the number of doses because it's a longer lasting immune response, and many times it can be taken orally. So no injection is required. And this might seem like, yeah, great, I don't like shots, but the bigger deal is in developing countries where you don't have, it decreases the cost if you don't have to have individual needles for everyone, and if you don't have to have a trained healthcare provider doing the injections. The negative is there is the potential to cause disease. Because the virus is replicating, you can sometimes get revertant mutations. So it reverts back, to that more virulent strain. Another negative is it requires cold storage. Okay. Not an issue here, a big issue if you're taking this vaccine to, say, Africa, where you may go to villages without any electricity. Another negative, positive, depending on who you talk to, is that infected people can shed live virus. So the negative is that someone that's not vaccinated has the potential of getting exposed to this virus. The positive is that the non-vaccinated person is exposed to this weak virus and it may pass on immunity to them. So kind of like a vaccination that you didn't ask for. The problem with this, with polio, is that polio is so stable in the environment that we're still finding live virus that if it reverts would cause disease in people. So that's attenuated, weakened, sometimes people call them weakened viruses. Okay. An inactivated one, the positive is that there's no risk for disease. Okay. There's no issues of contamination by other microbes, which I guess has sometimes happened in some of the attenuated vaccines due to the chemical heat treatment. Right, so it's a little cleaner. And it's very stable. So you don't have to store it at 4 degrees. It will have a longer shelf life, so that is an advantage for those types of vaccines. The negative is it's not as long lasting. Because all your immune system is doing is responding to the amount you get. It can't replicate, it can't create more virus for a longer lasting immune response. Okay, so therefore you're going to require more boosters, which means additional vaccinations. It must be injected. So that means you need needles and you need healthcare workers or someone trained to do the injections. You also don't get as full range of immunity. So remember, I am not an immunologist. But there are different types of, immune responses, your humoral, your mucosal, and so these inactivated ones don't seem to give you as broad of immunity. So in fact, inactivated ones don't seem to stimulate the mucosal immunity. So positive and negatives for each type. I wanted to give you a little example of attenuated and inactivated vaccine for the same virus and talk about the positives and negatives. So the oral poliovirus vaccine was called the Sabin vaccine. It was taken on a sugar cube, so no injections. It needed cold storage, so that was a negative. So I'm going to put negative, positive. Yeah, let's just erase this. That's just who it is. Another positive was that you got intestinal. So you got the mucosal immunity. I think that's right. I don't know. Intestinal immunity, which is where the virus normally replicates, because you were taking this orally. And polio is transmitted through the fecal-oral route, right? So you would ingest poliovirus, it replicates in the gut. The times it causes paralytic poliomyelitis is when it escapes from the gut and replicates in nerve cells and destroys them. The biggest negative was what we called vaccine-associated paralytic poliomyelitis. Vaccine-associated paralytic. So basically, vaccine-associated disease. Myelitis. I hope I spelled that right. So a small percent of people actually contracted polio due to the vaccine reverting to the neurotropic type. So they also developed an injectable vaccine called the Salk. Oh, so I didn't say. So this one was attenuated, the oral one. This one is inactivated. The negative was not as strong of protection. Darn it. You had to have needles, so you had to have injections. Oh, sorry. And you got bloodstream immunity, which is great. It's still protective, but you weren't getting that same intestinal immunity where if you actually were challenged with poliovirus, that's where it would first start replicating. The bonus of this was that it would protect neurons. Oh, I also forgot to put over here, negative, positive is that you shed live virus. Sorry about the handwriting. Which again can be positive in helping to spread that herd immunity, can be negative in that the polio is still hanging out in the environment. A really scary story about the inactivated is in 1955 there was one batch that they didn't completely inactivate, and that vaccine basically gave thousands of people polio. It was not good. Luckily we have, I think, stricter requirements on testing every batch of new vaccines made. Influenza, I'm just going to say one brief thing, and that our goal for influenza is a universal vaccine. And this would be more our, the goal here is to target a more stable area of the virus, so that something that's stable, meaning it's not going to mutate. So the issue with influenza is antigenic drift, which just means mutations. And that influenza can change its outside just enough so that the antibodies you have made previously no longer recognize, say, the spikes on the outside of the influenza, and now those antibodies don't protect you. And so you've got this delay in the very specific adaptive immune response, and that's why you end up getting sick. So we will read a paper on groups trying to design a universal vaccine that will allow us to have the same vaccine year after year for influenza. All right. Subunit vaccine means it's part of the virus, not the whole thing. Again, the same goal, stimulate an antibody response. The challenge is to figure out that right part of the virus that will make neutralizing antibodies. And we talked a little bit about neutralizing antibodies and how they are important for preventing infection. So for subunits, the positive is it's totally non-infectious. Okay? Because it's only part of the virus. It's not a whole virus. There's no genomic information. It's not even sometimes a whole capsid. So there's no replication at all. The negatives, is that it's very specific, which sounds like a positive, and it can be positive if the virus doesn't mutate. So if the circulating virus, we'll put this kind of between the two. If the circulating virus is stable, then these subunit vaccines work really well, like human papillomavirus. So this is a positive for HPV. If it's a virus that mutates, this isn't so great because then you'd have to reformulate the whole thing. Another negative is that it's limited to B-cell memory, antibody memory, because it's not infecting cells, and so that's usually where you get your T-cell response that addresses infected cells, and you're going to require multiple injections. Because it doesn't stimulate the immune system as well as a live attenuated virus. You're also going to have to add adjuvants, which are molecules that help stimulate the immune response. So sometimes these subunits on their own aren't enough to get a strong immune response, and so you have to tack some stuff onto them. Kind of related to subunit vaccines are virus-like particles. These are basically empty capsids. They self-assemble in vitro, so in the test tube. This is used for hepatitis B and HPV. So some HPV are considered subunits. Some have virus-like particles. I honestly can't remember which is which. They're looking at being able to do this for HIV and influenza, but the issue is, how do you get an envelope wrapped around these little capsids? Other terms you hear with vaccines is multivalent. So this can mean a couple things. It can mean either there's multiple strains of a single virus in one vaccine. So for instance, the flu is called quadrivalent because it's got H1N1, H3N2, and 2 of the B types of influenza. HPV is multivalent. It usually protects you against 4 different types of human papillomavirus. Or multivalent sometimes describes multiple, we'll just say viruses in one vaccine. Like your DTaP, which is diphtheria, tetanus, and pertussis, 3 different, protecting against 3 different organisms in one vaccine. The challenge whenever you start putting multiple things into a vaccine is making sure you're not getting an overreaction from, especially these, from the all the different all the different viruses or microbes in there. You don't want to stimulate an interferon reaction. So the reason they do this is to decrease the number of injections someone has to get. Current vaccines that we have, I just wanted to let you know. So MMR is measles, mumps, and rubella. And these are 3 attenuated viruses. In one vaccine. Polio. Most are using the injectable, which is the inactivated. But some countries are still using the oral, which is the attenuated, just because it gives a better immune response. Influenza we've talked about. The shot is inactivated. Sorry. Hepatitis A is an inactivated virus. Hepatitis B is a subunit vaccine. I'm sorry. Rotavirus is attenuated. Varicella, which is the chickenpox, shingles. Is attenuated. And that's why I said you can still get shingles even if you've been vaccinated for chickenpox. Human papillomavirus is a virus-like particle with capsid components from 4 strains. Some strains that cause warts, some strains that cause cancer. Always need to check if you're traveling. We've talked a little bit about military, or if you're coming into this country, sometimes you have to have different vaccinations. Yellow fever is an attenuated virus. It is one of the oldest vaccines, most safe vaccines out there. And the vaccine for rabies, you can get, it's an inactivated virus. And if you're a veterinarian or an animal handler, you can get a rabies vaccine as preventative. Most people do not get this. And so most people use it post-exposure. And so if someone's bit by a bat or a dog that's shown to have rabies, you will get this injection of the vaccine. Sometimes they give you an injection of antibodies, which we'll talk in a minute. But the whole idea is to race to get enough antibodies made before the virus gets into the nervous system and reaches the brain. So vaccines to eradicate diseases, there's basically 5 requirements. One, there has to be no reservoir, whether it's animal or the environment. Oops. Okay, so you can't have the virus hanging out somewhere. It makes it very hard to eradicate it. Oh, sorry. You have to be able to identify the infection. Right, so smallpox the only virus we've truly eradicated, gave a very distinctive symptoms of infection. You had these red blistering poxes all over the person. So if you can't identify the infection, then you can't be for sure that you've gotten rid of the virus. The virus has to produce stable antigens, right? So something that's ever changing is not going to make a good candidate for eradication because it's constantly changing. You need to have long-lasting immunity so that you can get everyone vaccinated and prevent future spread. And you have to have a virus that does not persist. We talked about persistent and latent infections. So if it persists or goes latent in the body, then you're not going to be able to eradicate it because it's going to be hanging out and it can pop back up. So smallpox had all of these characteristics. So smallpox only infects humans, so there was no outside reservoir. Like I said, you could easily identify infection. It is a stable virus, does not mutate. The vaccine we had gives long-lasting immunity, and the virus does not persist or go latent in the body. On the other hand, poliovirus has the issue of it does have an environmental reservoir. It can hang out in the sewage and the water and the soil. What else was I going to say? Oh, and polio, there is some evidence that it may persist in the body longer than we think. So there's ideas of reactivation of poliovirus. This is a little controversial. This is the main reason that we can't eradicate poliovirus. We talked about Andrew Wakefield and that he published some erroneous falsified data linking the MMR vaccine to autism, and that what happened is the incidences of measles and mumps increased because people were not getting the vaccine, trying to protect their children from autism. Again, his data was shown to be false, and he was charged with misbehavior to developmentally challenged children. He put them through these crazy tests just to try to prove this link between the MMR vaccine and autism, but unfortunately it's now in the minds of people that vaccines are bad. So how do we decide to vaccinate? It's kind of an interesting relationship. So when the risk of disease is great and the adverse reactions to the vaccine is low, the number of people vaccinated increases. When there's a very low disease risk, say West Nile virus, or the adverse reactions to the vaccines is greater than the risk of disease, the number of people vaccinated goes down. Okay. So right now in this country, we give people the choice to vaccinate or not. And it's something as a parent you have to decide about. So the challenges of the future for vaccines is how do we deal with ever-changing viruses? Right. How do we make that universal flu vaccine? If we're thinking globally, how do we distribute to war-torn countries or to isolated populations. And what goes along with that is, how do we overcome fears about vaccinations? There's fears in this country, there's fears in other countries, like, why are these Americans or Europeans sending over these shots to give us? There's distrust. There's not the same type of education about vaccines or the usefulness. Or I could just imagine it being flipped and another country sending medicine to us and saying, here, inject all your Americans with this. I bet there would be distrust here too. So there's hurdles. I doubt we're ever going to wipe out infectious disease. But I think this is the one that most research is dealing with tackling. How do we make vaccines that work, especially with viruses that continue to change? Okay, there's two more topics I want to talk to you briefly about. One is immunotherapy. So this is a temporary or passive immunity. And remember, whenever you hear the word immuno, you have to think antibodies. Okay. And so what you're doing is you're providing antibodies to a patient. They are not making antibodies themselves. So this is not really stimulating the immune system or any kind of memory. This is kind of a quick fix, or a fix for people who might be immunocompromised. So the idea is that you make neutralizing antibodies. You give an injection of these antibodies to the person. The problem is that it's short-lived. Like, they say about 30 days the antibodies last before they're degraded. The positive, I'm not going to say this is a negative, but just a bullet point. The positive is its immediate protection. Wow. I have no idea why my pen does that. Right, so you don't have to wait for your B cells to be stimulated to produce these antibodies. It's immediate protection. Okay, so who would get this? Infants that get sick. The reason breastfeeding is so good, besides nutrition, is that the mother is providing antibodies to the newborn. So it's passive immunity, helping that newborn prevent infection based off the mother's antibody production. But immunocompromised people, such as people getting a transplant, going through maybe chemotherapy, or if you're born with an immunodeficiency, immunotherapy can be a positive to help, again, prevent a devastating illness. The one issue they have to worry about is, will your immune response react to the antibody and see the antibody as foreign and then try to destroy that antibody? If that's the case, then the antibody won't work again. So that's what immunotherapy is. The other thing I want to just mention briefly, and this is something you're looking at in homework 3, is antivirals. So antivirals is stopping the viral infection. And so let me write here. You must target something specific, unique, to your virus. Okay. Most of the time, it targets a viral protein or a viral enzyme, which are proteins. Okay. The issue, so here's some antivirals for different viruses. The issue is mutation that leads to resistance. So with something like HIV, they actually give people what we call a cocktail, so a mix of multiple antivirals to try to knock down the virus level before the virus has a chance to mutate and resist or become resistant to those antivirals. They found a long time ago that using only one antiviral, the virus would mutate rapidly enough to be resistant to that antiviral, and then they would try another one, and then they'd get resistant to that one. And so the cocktail mixture seems to work a lot better. All right, so those are some of the ways we deal with trying to prevent and to deal with virus infections. Thank you.